I just wanted to say one quick note before we get started, and that is that if you're online, please use the raise hand function or write a question in chat if you have a question or a comment. We'll see how much time we have for questions at the end. I know there was one mailed in that Nora may have already gotten, so we'll see. Anyway, so I'll go ahead and get started because I think we're pretty much where we're going to be. And I have a few remarks to make. First of all, happy Leap Day. It might be appropriate that we're celebrating Bud on Leap Day. Maybe we'll do it 4 years later. No. We'll try to do it every year. So anyway, I'd like to welcome you to the first annual Richard Bud Veech Memorial Lecture. I also want to welcome members of the Veech family, his daughter Jennifer, who is here somewhere, okay, and son George, who should be watching remotely. We appreciate all that your father did for and at NIAAA. I know personally he was a great inspiration to me scientifically. I'd like to thank the many NIAAA faculty and staff members who contributed their ideas and support for this lecture. I can't name them all, but George Kunosh, Michelle Antoine, Steve Vogel, and Todd King were really instrumental in helping us formulate the idea to do this and then starting to get it together. I really need to thank Emily Surratt, David Montgomery, and Todd King for planning and logistical support. And special thanks to Kara Breeden and the communications branch for helping us make the flyers and get the information out along with Emily. So I really appreciate that. And finally, I also want to thank the NIH events staff here in the Porter building for their help with this venue and giving me some tips about how to do things. So I hope everything goes seamlessly. So Dr. George Kunosh, the former scientific director of the Division of Intramural Clinical and Biological Research at NIAAA, couldn't make it today, unfortunately. And he was originally scheduled to give an overview of the life and accomplishments of Dr. Veech. So I'm going to have to fill in. But what I'm going to do is pretty much read what George Kunosh sent me. So bear with me, it might be a little wooden. So Dr. Richard Veech, a giant in the field of biochemistry of energy metabolism, had been a longtime member of the intramural research program of NIAAA when he unfortunately died 4 years ago at the age of 84. Bud, as people fondly called him, grew up in Illinois, where he still owned and managed the family farm at the time of his death. He went to Harvard to first earn an undergraduate degree in history and literature, and then a medical degree in 1961, where, as I understand it, he was a classmate of former NIAAA director T.K. Lee. That was followed by a clinical residence in surgery. He subsequently did graduate studies at Oxford in the lab of the Nobel laureate Hans Krebs, the Krebs cycle guy. And he earned a Doctor of Philosophy there in 1969. That is where he had acquired a lifelong interest in cellular energy metabolism and its implications for the treatment of human disease. Early in his career, he discovered that the toxicity associated with hemoglobin-based oxygen carriers used in resuscitation fluids was due to lactate in the Ringer solution, used as an excipient, which promoted the generation of oxygen-free radicals. So he came up with the idea of replacing lactate with beta-hydroxybutyrate, and this eliminated the toxicity of lactate, and Veech's modified resuscitation fluid has been widely adopted by emergency rooms worldwide. During the last 3 decades of his life, Bud Veech focused on the therapeutic potential of ketones, and he became a virtual godfather of this rapidly growing field. Veech realized that ketones are not simply toxic byproducts of diabetic ketoacidosis or starvation, but can serve as alternative energy sources when glucose becomes unavailable, such as in insulin resistance. He explored the multiple mechanisms by which ketones serve as efficient sources of ATP generation, some of which I think we'll hear about today, improve tissue blood flow in hypoxic conditions, and exert anti-inflammatory effects. With an eye on the practical implications of his work in treating numerous diseases of defective cellular energy metabolism, including epilepsy, Parkinson's, Alzheimer's disease, Huntington's disease, and type 1 and 2 diabetes, he developed an inexpensive methodology to mass produce ketone monoesters in an edible form, thanks to Todd King, who's also. As a true physician and humanitarian, he placed helping sick people ahead of financial gains for himself, and that is really true. He educated people on the value of eating low carbohydrate, higher fat diets, overnight fasting, exercising, or otherwise increasing the generation of ketones by the body itself. His true humanitarian nature was well exemplified by an incident in the early 1970s when he was only one of 10 survivors in a plane crash in New Hampshire that killed 42 people. Even though he was seriously injured himself, he was busy helping other survivors when the first responders arrived. Although Dr. Veech is no longer with us to witness the growing acceptance of his ideas, his impact on the field and more broadly on human health and well-being will remain with us. We will also remember him as a caring human being and one with a wry wit. When interviewed once about the epidemic growth of obesity and addictive disorders, he famously said, and I might throw in a Veech voice here, "People like to eat junk food, drink a lot of alcohol, sit on the couch instead of exercising, and want a pill to help them stay healthy and fit. Well, good luck." So with that, I'll turn the podium over to Dr. George Koob, the director of NIAAA, to give the introduction for today's speakers.
Well, I can't match that one. So it's a real pleasure today to introduce our two speakers, Dr. Nora Volkow and Dr. Corinde Wiers. I've known both of them. I've collaborated with both of them. Nora's one of my best friends and colleagues. She's the director, as many of you already know, of the National Institute on Drug Abuse. She's been director since 2003. I just celebrated my 10th anniversary at NIAAA, and I'm trying to catch up with Nora, but I don't think I'm ever going to get there. Her work has been instrumental in demonstrating that drug addiction is a brain disorder, a conceptual framework, of course, that I completely support. And as a research psychiatrist, Nora has pioneered the use of brain imaging to investigate how substance use affects brain function. Her studies have documented how changes in the dopamine system alter the function of brain regions involved in reward and the self-control that's associated with addiction. She's also made important contributions to the neurobiology of obesity, ADHD, and aging. As many of you know, she was born in Mexico and earned her medical degree from the National University of Mexico in Mexico City, where she received the Robbins Award for being the best medical student of her generation. Her psychiatric residency was at New York University, where she earned a Laughlin Fellowship from the American College of Psychiatrists as one of the 10 outstanding psychiatric residents in the United States. So Nora had an outstanding career from early part of her career. She spent a good bit of her time at the Department of Energy's Brookhaven National Laboratory in New York, where she held a number of leadership positions. Dr. Volkow has published over 1,000 papers, peer-reviewed articles, 113 book chapters, manuscripts, and co-edited Neuroscience in the 21st Century. And she's also edited a number of books on neuroscience and brain imaging and substance use disorder. She's won all kinds of awards, including the Nathan Davis Award for Outstanding Government Service. She was a Samuel L. Heyman Service to America Medal finalist, and she's a member of the National Academy of Medicine and the Association of American Physicians. There's a number of other awards that Nora has won, but I looked through her CV and I noted that she has been honored 11 times with honorary doctorates from 11 different institutions across the world. That is an extraordinary accomplishment and a testimony to how highly valued she is by her peers. So in short, you can read about some of the details in the booklet. Nora is a rock star in the addiction field. And if you don't believe me, just go to a meeting with her and you'll find out, okay? It's really hard to get a word in edgewise or get a quiet place where we can talk about something that we need to talk about with NIH. I know her to be a person with the most penetrating and erudite questions about addiction, and among those, among all my colleagues. So it could possibly strike terror in your heart if she's asking you a question, but actually, when you think about the question that Nora asked you afterward, you realize it opens up a whole new venue of way of thinking about something. So I'm really happy, Nora, that you were willing to participate in the first Richard L. Bud Veech Memorial Lecture. Now, Corinde Wiers is an assistant professor of psychiatry and radiology at the Perelman School of Medicine at the University of Pennsylvania. She received her Ph.D. in psychology from the Berlin School of Mind and Brain and the Free Universität of Berlin. She completed a postdoctoral fellowship at the National Institute on Alcohol Abuse and Alcoholism in the Laboratory of Neuroimaging, that is directed by Nora Volkow. As most of you know, Nora has her laboratory in NIAAA, and I have my laboratory in NIDA. They do that to keep us from getting in trouble, but we get in trouble anyway. She was mentored by Nora Volkow in her postdoctoral fellowship, and there she studied neurocognitive underpinnings of substance use disorders using PET and MRI. It was here in her work with Nora that she began her work on the effects of ketogenic diet, on alcohol responses, and in particular on aspects of alcohol craving in the broad sense of the word. She currently investigates the effects of nutritional ketosis on brain energetics, alcohol withdrawal, alcohol consumption in individuals with alcohol use disorder, and I'm firmly convinced that Corinde is going to be our next rock star. So thank you also, Corinde, for participating in this memorial lecture. I want to say one thing about Bud Veech. I only encountered Bud a couple of times at major events at NIAAA, but I heard more stories about Bud Veech than anybody else in the whole institute. Bud was very much way ahead of his time, and we now are recognizing more and more that alcohol use disorder is not only a brain disorder, but it's a body disorder. And with that last sentence, Nora, it's all yours.
Good afternoon, everyone, and it's an honor for me to be here, and that was an extremely kind words, George, and it's, for me, has been a pleasure, and I would say some of the most exciting projects that I've been able to push because we're doing them jointly. So it has been an extraordinary partnership, not just as researchers, but I also think very importantly as directors of two institutes. And hopefully this will continue and inspire others about the importance of bringing and coming together. It's also an honor for me to also be speaking with Dr. Corinde Wiers, because it does epitomize that way that science creates. I mean, it's part of it's a discipline that allows us to actually improve humankind in general and to accumulate knowledge. And so there has to be this transition and movements across the generation. So it's wonderful to see Corinde taking off and running with the potential that understanding the metabolic effects of alcohol has, not just an understanding, as George was saying, that it's a body disease, but very importantly, what is that component that is related to the reinforcing effects of alcohol? And this is something that has always intrigued me because of the drugs that can produce addiction. The only one, unless you are eating some of those brownies over there and they contain THC, but I'm sure they don't. But other than that, there are no energetics, which also makes me feel very guilty. They were giving this presentation of Dr. Veech and said, don't eat carbohydrates, eat something more nutritious. And I just had for lunch a cookie and a brownie. So, and I'm trusting the brownie. So if I am too wired today, it is because of the brownie. So it is a pleasure to be here in a serious note. And it does reflect. And so what I'm going to be speaking to you is work that I had started to do actually a little bit before at the end of the 90s, towards the beginning of the 2000s. where I was very much interested on using brain imaging to actually by pharmacologically challenging individuals with different types of drugs, including alcohol, to try to understand what were the areas of the brain involved with the intoxication, number one. But number two, something that I think it is fascinating and a powerful tool that we have. If any one of us is given a drug, each one of us is going to be responding to it differently. And that different response is a function not just on the context in which we're getting the drug, but importantly into our biology. So to me, the ability to challenge the system and extract information provides a means of understanding what may be underlying neurobiological processes. And so that's what led me many years ago to start to systematically use positron emission tomography at the time that we didn't have MRI to investigate the effects of cocaine, alcohol, as well as stimulant drugs that we use for the treatment of ADHD. So today I'm going to be speaking specifically about alcohol because this is a field where I took the scientific questions, but was very much guided by Dr. Veech and also by Dr. T.K. Lee. So where did it all start? It started again, I was saying I was looking at doing acute alcohol administration on people. We were using initially very high doses of alcohol, 1 gram per kilogram. And we were observing very, very paradoxical effects. We were observing that regardless of the doses of alcohol that we were given, there was a very dramatic reduction in brain glucose metabolism. And it didn't really completely make sense because in parallel, if you do studies of cerebral blood flow, you do not observe that reduction in cerebral blood flow. And the other thing that was particularly paradoxical in my brain, I always say when I see these things, it just hurts my anterior cingulate cortex, was that we were seeing, you know, if you give alcohol to a person that has an alcohol use disorder, they are extremely tolerant. So you can give them much higher doses without them feeling intoxicated. So we did a study, and at Brookhaven National Laboratory, they allow us to give high doses of alcohol to individuals with alcohol use disorder. So we compared the responses of individuals with alcohol use disorder and normal controls. And to my bedazzlement, I don't know if that word exists, but to my bedazzlement, the individuals with alcohol use disorder, at that time we called them alcoholics, had a dramatic reduction in brain glucose metabolism, dramatically. Their brain looked actually as in the severe stages of Alzheimer's disease, but they were not reporting feeling of intoxicated. And the effects were much larger than healthy controls in which a 1 gram per kilogram of alcohol made extremely intoxicated. So what it was clear in my brain, because obviously it would have been a very nice story to see an association between the reductions in glucose metabolism and the behavioral effects of alcohol, because then it would have allowed us to say we can use glucose metabolism as a marker of brain function. But if you don't see any effects despite these dramatic reductions in glucose metabolism, that raised a gigantic question in my brain. So I couldn't let go of it until I understood it better. So we did multiple studies, and all of them systematically showed that the alcohol in healthy controls, much more than in the individuals with alcohol use disorder, produces significant reductions in brain glucose metabolism. And this is one of the studies actually just showing 2 different doses of 0.25 grams per kilogram versus 0.5, and you can see that dose-related effects. But even at the relatively low dose of alcohol, you see very significant reductions in metabolism. And because it's not at all associated with any subjective effect or sleepiness or drowsiness or anything that we could figure out, that led me to force me to think this is not reflecting reduction in brain activity as I had initially interpreted these findings. And certainly I was interpreting the findings that you get when you give a benzodiazepine drug that also decreases metabolism, but where that decrease is linked with sleepiness and reduced alertness. So we then constructed I'm going to be speaking to you about 3 studies that are linked. And I do believe very strongly that in science you need to replicate because our studies, certainly with brain imaging, recruit relatively small number of subjects. So whatever results you get, you do not know that they are necessarily representative of the general population. So this was the last of the studies with this characteristic where we actually investigated the effects of acute alcohol. Again, it was 0.75 grams per kilogram in this very simple design, 2 FDGs, one, you give them a drink and you try to make it as placebo possible as alcohol as you can, and the other one you get the alcohol. And then you look at what you are observing. I'm going to present you the data on 15 healthy controls, 12 of whom were males. Again, we replicated. This is one of the most reproducible findings. Decreases in brain glucose metabolism, not associated with actually anything that we could measure. But the issue that I also wanted to do in this particular study was I was trying to understand, are there areas of the brain that are more sensitive than others to that reduction in the metabolic effects of alcohol? Or is it just an homogeneous pattern? And from prior studies, it appeared that there were some areas that were actually more decreased than others, and others were minimally decreased, if any. And in this analysis for this particular last study that I'm speaking about, what you're seeing there are highlighted the areas of the brain where alcohol produced the most dramatic reductions in brain glucose metabolism. And I would say, that reflects the occipital cortex and the cerebellum. And that is also very reproducible finding. Because these are all slides, and we didn't have the very fancy tools that we currently have for display data. I am not presenting the subcortical structures, specifically the striatum and thalamus, in the images themselves. But you can see that actually, if you were to look overall at what are the areas that are less sensitive to these reductions in absolute metabolism, we are measuring how much it's reducing consumption of glucose per minute, is the thalamus and the striatum, whereas the areas with the largest changes are the occipital cortex and the cerebellum. So it is not homogeneous. There are significant differences in the cortex, but the largest differences relate to the striatum, the globus pallidus, and the thalamus, and certainly the cerebellum being a region with very high sensitivity. And so then we ask the question, well, what about individual variability? And I love to look at individual variability, particularly if you are looking at small samples, you cannot not look at them. So I'm showing you individual data for the 15 subjects under the metabolic measures as placebo and alcohol for the different areas of the brain, the cerebellum and the occipital cortex, as you see here. And what you can note, if you see the placebo is in dark brown, and then the alcohol is in the empty circle. Look at the subjects on cerebellum and occipital cortex. Look at occipital cortex, how consistently there is a reduction on every single subject. Now, if you look at the other regions like thalamus and striatum, let's look at, pay attention, for example, this one is striatum. You actually see what you don't see in the cortical regions. There are some subjects in which the metabolism was higher during alcohol intoxication than during actually the placebo. Begging the question about what is driving these differences between individuals in the responses overall in the magnitude of these decreases in brain glucose metabolism. And I think that this is an extremely important question for which we do not have specific answers. We have been measuring plasma concentration of alcohol to see if that was driving it, and it's not. So we concluded, yes, there is a significant decrease in brain glucose metabolism during acute alcohol administration that was not, like in the other studies that we have done, associated with behavioral effects. So why is that so? What is it telling us? And this item that I just highlighted, we also show significant variability in effects of alcohol between subjects. So here jumps into the second study. So what's going on? And at this time, I was starting to speak with TK Lee, and he said, Nora, you need to speak with Veech, with Richard Veech, because he is the expert. I was discussing with TK, is it possible that what we are doing is shifting the energy substrate in the alcoholics, which could, because they are constantly exposed to alcohol, they may have the enzymatic advantage to transform alcohol into acetate, which we know is driven by two enzymes. Alcohol, the dehydrogenase, and acetal, the dehydrogenases. And acetate then is at the end of that run. And it's also clear from multiple studies that have been done in the past that people that are alcoholics, and I'm going to use that term, I don't use it in any way pejoratively, but that's the way that we used to describe, and that's also the way that's in the literature. And so they show significantly higher concentrations of acetate in the plasma. And that sort of highest when you're intoxicated and with time, but they can, levels of acetate can be actually elevated for a couple of days after the person stops taking alcohol. So we're discussing this with TK and said, Nora, and it was TK, the one that said to me, Nora, I think that what you are doing is you're substituting acetate. Acetate could be the reason why these individuals, when they are intoxicated, are not consuming glucose. So speak with Veech. And so we did have several meetings with Veech, and out of those discussions, he actually did a study that documented in rodents that effectively alcohol produced acetate that then was used for energy substrate. And this is indeed known, and actually glucose goes through two processes. The first one is the glycolysis, and glycolysis is a way of very rapidly extract energy from glucose, but it's very inefficient. You just get 2 ATPs. And then it goes into the Krebs cycle, that one that Richard Veech studied. So the Krebs cycle is a very efficient system and provides approximately 36 ATPs. So you have in the brain, in brain cells, in neurons and glia, the ability to use glucose both of these ways. And right now, the consensus is that the Krebs cycle, the utilization of oxidative metabolism, which is the one from the Krebs cycle, is used as the main source of glucose to sustain the energetics that are necessary for all of the constant processes the brain has to maintain in order for example, to sustain the electrical gradient. The brain consumes huge amounts of glucose, needs enormous amounts of energetics. Whereas the idea right now is, on the other hand, that glycogenesis, which provides very fast ATP, is a mechanism that you can invoke when you need to actually generate a very fast response. So then with these discussions and a strong push, I mean, TK was saying, Nora, yes, and Richard Veech had other ideas of he was saying, well, maybe hydroxybutyrate could be a source. So I basically bet on acetate, and we decided then to do a study to compare the effects of alcohol intoxication on glucose metabolism on the one hand, which we already knew what we were going to see, but on the other one to look at acetate metabolism. And I was very, very fortunate to have Dr. Sonny Kim, who I think is back there, who actually labeled the acetate, and to just say to Sonny, I need a ligand, and there we could do these things very rapidly. And you say, easy, don't worry, I'll get it to you. And immediately got the synthesis. So we had C11 acetate, which allows us to use positron emission tomography to evaluate how alcohol affected acetate metabolism. So the study was done into groups of subjects, 60 occasional social drinkers, and 15 heavy drinkers. They did not allow us at that point then to actually recruit individuals with an alcohol use disorder. At that time, it became much more restricted to give alcohol to individuals with a severe alcohol use disorder. So, but they were very heavy drinkers, and we used C11 acetate, again, to measure the effects under two conditions, placebo and alcohol intoxication. The dose of alcohol that we gave is the one that we use for most of our studies at Brookhaven, 0.75 grams per kilogram. And what we observed first was there were no differences in the behavioral effects of alcohol between people that were regular users, heavy users, and the occasional users in terms of high, intoxication, sleepiness, dizziness. But where there were differences were the self-reports of liking. People with heavy drinking when they were exposed to that dose of alcohol in the laboratory, which is not ideal because you are sort of crucified like that in the PET scanner, right? So it's not the most conducive to these euphoric responses. They still like it very much, and also found it significantly more pleasant. So there is a distinguishing effect in terms of the quality of the reinforcer in people that are regularly using it. What about acetate? And first, before we go into acetate metabolism, let's look at that whole extremely important question. If you are a regular drinker and consume high quantities, the concept is, are you able to metabolize alcohol faster? And if you do not control for that, of course, all of your findings are going to be confounded. So we observed there were absolutely no differences in the concentration of alcohol in plasma between the occasional social drinkers and the heavy drinkers. All of the data I show you are standard deviations. It's not error bars. All of them, standard deviations. And then onto your right, you are seeing the plasma concentration of the radioligand, which is C11 acetate, because also if there are differences, for example, in radiotracer delivery and metabolism of your radiotracer, you need to account for that. So there were absolutely no significant differences in anything. So what about C11 acetate metabolism? So here you have the images from two subjects. On the top one, you have an occasional social drinker. To your left is the placebo, to your right is the alcohol. And the lower one is the heavy drinker. And the colors to the scale shows you the changes, the concentration, the uptake of C11 acetate, which is used as a surrogate of acetate metabolism. And what you can see right away is that alcohol, people when are intoxicated with alcohol have an increase in C11 acetate metabolism. Parallel, they have a decrease in brain glucose metabolism. What is also clear from this one, in this particular subject, is that overall it appears like the heavy drinker definitively has a higher, for this particular subject, metabolism in the brain with C11 acetate during alcohol intoxication than the occasional social drinker. And it also seems like it's also occurring in the placebo situation. And just to your right, for those that are PET aficionados, we have the time activity curve for the C11 acetate, and you can see that, yes, there is significantly higher values of C11 acetate during alcohol than placebo, consistent with what you are observing here on the alcohol images themselves. So where all of the areas of the brain significantly observing the same effect increases. We observe significant increases basically in C11 acetate during alcohol administration for all of the brain regions. But there were some areas where it was larger. And the cerebellum was one of those regions where the effects were larger, whereas the thalamus was one of those regions where the effects were very minimal. And the question was then, are there differences between heavy drinkers and occasional drinkers? If you look at all of the regions, and you sort of actually, there were not so many regions because we didn't have the tools to do that very comprehensive analysis. So we just extracted 7 large regions, and you see, if you look at all of them, repeated measure, it is significantly different between the groups. If you say, is there any one region that differs between the groups? The one that reaches trend significance at 0.06 is the cerebellum, where you can actually see what appears to be higher metabolism, both in placebo and alcohol. So with this study, we actually showed that brain acetate metabolism was increased with acute alcohol administration, consistent with our hypothesis that during intoxication, the brain uses acetate as energy substrate. Acetate metabolism tended to be higher in heavy drinkers than in occasional social drinkers. And obviously the question is, what is driving this change in the sensitivity to acetate in people that are drinking regularly? And there are a lot of hypotheses out there in the scientific literature, including an upregulator of the transporters, the monocarboxyl transporters that are necessary for acetate to get into the cells. And that is likely to be one of them. But another hypothesis is that the enzymes that are needed for acetate metabolism and entry into the Krebs cycle are upregulated in individuals that are exposed regularly to alcohol. So this is actually, and these findings were the ones that immediately, we discussed it in the paper, led us to think that sort of, if you are going to alcohol withdrawal, that was my first concern, you're going to be in an energy deficit because your brain has modified itself to maximize the utilization of acetate as an energy substrate. And those, when you go into alcohol, you'll be in energetic deprivation. So what I wrote as a concern was the notion that this could be toxic to the brain. And so I put forward the notion of, could you give alternatives to increase acetate in the plasma and so protect individuals for the negative effects? And that's what led to the concept of the ketogenic diet that became a reality finally when Corinde, because she's a force of nature, came in and got it done. And she was speaking about, because that's a very natural follow-up experiment that happened from it. So just to the last experiment related to that point that I was saying, when you consume alcohol, when you consume basically alcohol, you decrease glucose metabolism. But you are decreasing, I mean, the question is which pattern of the metabolism of glucose are you decreasing? By showing that acetate takes over, which goes through the Krebs cycle, the hypothesis therefore would be that the reductions in glucose metabolism with alcohol are predominantly driven by oxidative metabolism, but that alcohol should not affect the glycolysis component. So in order to test that hypothesis, that we would basically see actually differences in the effects of alcohol on glucose metabolism that is related to just energetic tone, not stimulation, versus differences on the effects of alcohol when you challenge the system with the idea that it will require glycolysis to process the information. So that's what we did. We did a very complicated study that actually was done in 23 healthy controls and 25 heavy drinkers, because we were trying to see if there were differences, again, with brain glucose metabolism. We did FDG. Each subject was scanned 4 different times on 4 different days. And I want to tell you something too, because this study would have never, ever been possible if it were not for Dr. Jean-Jacques Wang, who actually has been the lead clinician at the laboratory and who's able to perform the most complex experiments and get everybody to work together. So 4 different FDGs, the subjects had to come at different times, of course. But the dose of alcohol was always the same, 0.75. So what was the difference? Look at it in 2 pairs. In one of them, they have a placebo with looking at the screen that doesn't have anything. In the other one, they have a placebo with visual stimulation, a video. They are presented with a video. And the other set is the same experiment, but they are intoxicated. So we give them alcohol, 0.75 grams, then we put them into an empty screen, and on another day, they have the video. And we compare the response with the hypothesis that the main differences are going to happen when there's no stimulation, whereas the magnitude of the changes in the metabolism brought about by the stimulation would be equivalent. And then of course we compare differences between heavy drinkers and non-drinkers. And there were no differences like we had seen in the past for example high intoxication, but just for desire. And the desire for alcohol was actually higher, whether they were placebo or alcohol. So they had just higher desire. There were no differences in the concentration of alcohol between both groups. This is just a summary for if you take all of the data that we had of the 4 FDGs and we ask the subject, what's the difference of this one for between heavy drinkers and normals for the placebo condition, eliminated all of the visual stimulation, just the placebo condition, whether you were, you see that significant reductions in glucose metabolism with alcohol intoxication. And those reductions, just like we had seen many years ago, 2 decades ago, were higher for normals than heavy drinkers. So yes, heavy drinkers have an attenuated glucose metabolic reduction when you are giving them alcohol. And then we did visual stimulation. So this is the data to show when you do visual stimulation on placebo in the first row versus alcohol on the lower row, which areas of the brain show the most activation, which is what this data shows on glucose metabolism. And you can see what you expect, the visual cortex, Brodmann area 18, but you also see the auditory cortices, which actually are the Brodmann corresponding to Brodmann area 22, because it was a visually narrated video. So this is not surprising, and you can see it actually, whether it is placebo or alcohol. If you compare the placebo versus the alcohol, you can seem to see, and this is both for the normals and the heavy drinkers, that it appears to be, for this comparison, a larger effect under the effects of alcohol intoxication. And this is a summary which actually shows the effects of the brain on the 4 conditions. You have the placebo more than alcohol on the heavy drinkers on the top, follow the placebo more than alcohol on the normal non-heavy drinkers, and then on their visual stimulation, placebo more than alcohol with visual stimulation in the heavy drinkers, and placebo more than alcohol in the normal for visual stimulation. Just look at the dramatic difference in reduction in brain glucose metabolism between the normal, the placebo, and the alcohol in the heavy drinkers, the first and the third row. And the equivalent for the non-heavy drinkers is the second and the fourth. So it is a very, very large effect that drives individuals with alcohol use disorder to be more sensitive. The other issue that is apparent in these images is actually that the visual stimulation effects decrease. And you can see that both on the heavy drinkers in the first and the third, the magnitude of the reductions of the third is less than the magnitude of the reductions on the first. And the same thing, if you look at the magnitude of the reductions in the occasional drinkers in the second, which predominantly goes to occipital and cerebellum, is less when you stimulate them visually. So, and this is likely to reflect that you are doing visual stimulation that drives energetics in order to respond to that particular stimuli. This actually just identifies the regions that have the normalized. I normalize the values just to highlight the differences. And you see exactly the same for alcohol, heavy drinkers versus occasional drinkers. For all of them, whether they are under the effects of alcohol or placebo, under both conditions, you see the activation of the visual cortex and you see the activation of the auditory cortex. And the magnitude of that activation did not differ for heavy drinkers and for normal controls, indicating that that process of energetics is not affected by the increases in acetate by alcohol itself. So how do we interpret this? We interpret this to indicate that alcohol basically can serve as an energy substrate, and that acetate is likely to be one of the main sources of energy during intoxication, and that acetate is predominantly going through the Krebs cycle. And therefore, it's actually taking over the ability of glucose to go through the same cycle. It is, as Dr. Richard Veech was saying, it's a very effective energy source that has generated a lot of interest for the treatment of many neurological diseases. The problem being is when someone is repeatedly exposed to alcohol and their brain shifts towards acetate metabolism, there are these periods of intoxication and then withdrawal. And during those states of energy deprivation, they're likely to be much more vulnerable. When we were doing these experiments, there was a lot of belief that the main cells in the brain that consume acetate were the glial cells. And there is a lot of preclinical work to indicate that they definitively are likely to be, they tend to favor acetate over glucose if the concentration is high enough. But it is now clear also that neurons also can utilize acetate. And so what we are observing now is, and that's the way that we interpret it, in heavy drinkers, there is a vulnerability that expresses itself as the acetate levels in plasma goes down, that actually drives the withdrawal, and that may be contributing to some of the neurotoxicity. Ergo, why we basically hypothesize that a ketogenic diet would be beneficial for improving during the withdrawal stage, but hopefully also to minimize the adverse toxic effects of alcohol. And so yes, there are two energy sources. You can go to your gas station and say if you're sober or drunk, and accordingly pick up one or the other. But I do want to end my presentation with the LNI team, because obviously this work is extraordinarily complex and involves an enormous amount of people to make this happen. I did mention Dr. Sonny Key, who labeled as 11 acetate. I mentioned Dr. Jin-Jack Wang, who actually has been carrying all of the clinical studies. I also have Barry Demiral, who's here, who also has been responsible for all of the management of the data and very much implicated in analysis. And Darlo Tomasi, who is our physicist. And we also have, obviously Corinde that came and make this a reality at a period where we have been trying to make it happen, but the complexity actually was intimidating many. but nothing ever intimidates Corinde, so she succeeded in doing it, and it's quite wonderful to see how successful she has been in building up the program at UPenn, and also, importantly, taking that responsibility that we all have of actually taking knowledge and bringing it into potential therapeutics. I do want to also, because I actually, in looking at this picture, recognize the help from Dr. Ethan Shohori, who's actually, I think I saw him there, who also participated in the data analysis. So thanks very much for your attention.
Now I need to switch the slides. Can the people online actually see your slides? Oh, and I see. But now it's not presented here anymore. Oh dear. Oh dear. It's in presentation mode. Uh-oh. Is somebody here well equipped with Zoom? Here, look. If I...
Yeah, I think we have to unshare.
Unshare this. Oh, actually, it didn't share it with you. Wonderful. All right. Well, thank you so much, Dr. Lovinger and Dr. Koop, for inviting me today. It's an enormous pleasure, and I just told Dr. Koop that it's been the best night of sleep I've had in a while. My 4-month-old baby is at home with my husband, and I was able to sleep here in Bethesda in a hotel. And yeah, also thank you, Nora Volkow, for continuous mentorship in the last 10 years. 10 years ago, I was jumping from joy and jumped on a plane to become a postdoc here at the NIH, NIAAA, under the mentorship of Nora Volkow, and yeah, primarily worked on the effects of a ketogenic diet on alcohol or alcohol withdrawal in the alcohol inpatient center here at the NIH. And it was in 2018 that we reached out to Dr. Veech to ask, well, could we actually do this study also with your ketone ester? And he invited me over, and it was incredible. Dr. Veech did not do half-an-hour meetings, 1-hour meetings. Nope, we sat there for a full day and discussed research ideas. And he either said, like, yes, that's definitely worthwhile, or no, Dr. Wiers, you may as well pick your nose, do not do that. And so he was incredibly honest. He had a desk full of papers that range from bright white to somewhat yellow at the bottom, and sometimes was able to just give me a paper here and there. Read that, definitely. But yeah, I will present primarily the studies that we've done over the past years on ketone treatment in alcohol use disorder. And I just wanted to add a few numbers on alcohol use disorder. That worldwide, 3 million deaths every year result from harmful alcohol use, and over 1 in 7 Americans are estimated to develop an alcohol use disorder in their life. And surprisingly, there are really good treatments for alcohol use disorder, FDA and behavioral treatments, but apparently only less than 8% of adults who need treatment for alcohol use disorder actually receive them. And there are various reasons for it, but stigma is one of them. And so overall, there's a need for interventions that can promote long-term abstinence and reduce alcohol craving. And what we've seen from Nora, I'll just recap it one more time. If you drink alcohol, glucose metabolism goes down, acetate metabolism goes up. And in people who are heavy drinkers or with an alcohol use disorder, acetate metabolism also during sobriety is up, whereas glucose metabolism is lower compared to social drinkers or non-drinkers. And this is relevant for alcohol use disorder treatment in that we hypothesize that if you quit drinking when you're detoxing, then this brain state of high acetate goes to low acetate, and there may be a delayed transition of the brain from low glucose back to high glucose utilization that could contribute or lead to these withdrawal symptoms in alcohol craving and relapse in alcohol use disorder patients. And a ketogenic diet is a diet that's very high in fat, low in carbohydrates, and it's frequently used for the reduction of seizures and epilepsy. That's been known for over 100 years now. They just celebrated the 100 years anniversary of ketogenic diet for epilepsy. And in this nutritional state of ketosis, the cells are using ketone bodies for fuel rather than glucose. These ketone bodies are acetoacetate, beta-hydroxybutyrate, and acetone. And the goal of the next studies and also of my lab at Penn now is well, could ketones be a replacement for acetate? So could it replace this hypothesized energy or help this hypothesized energy deficient state in alcohol use disorder? And one study from Denmark was a preclinical study where Nora was also involved, found that a ketogenic diet reduced alcohol withdrawals in rats. So it reduced body rigidity and irritability when rats were detoxing of alcohol. And we later found a paper from 1981 that actually showed something very, very similar. So sometimes we have to replicate things just because we don't remember that it was done before. But this is really, our preclinical model that we based our human study on. And at the NIAAA here on campus, we then performed this 3-week ketogenic diet trial in patients who were coming in here for alcohol detoxification. And it was very, very important to us that the study was blinded. But it's very difficult to blind nutritional intervention. You get spaghetti or you have to eat oil. So, right? Or do you eat a cookie or not? So we decided to give people a largely liquid diet. So patients received shakes for breakfast, lunch, and dinner. And in addition, they received snacks because they still wanted to chew on something. And all the snacks were ketogenic, so that's how we balance these two different interventions. And we used a 4-to-1 ratio of grams of fat to grams of carbohydrates and protein combined. And it's an extremely rigid ketogenic diet, but participants tolerated it surprisingly well. And what you can see here in the graph is more proof of principle that every day an independent person was looking at urine acetoacetate, that's graph A, and B, every week we took some finger sticks for beta-hydroxybutyrate. And you can see that both acetoacetate and beta-hydroxybutyrate increase in the ketogenic diet group compared to the control group. And there was a small dip in week 3. I think there are 2 participants who went on a pizza party that apparently is regularly organized. Well, we gave them a warning and they stayed in this study. Never did that. But it messed with our memes a little bit. And with the help of Jan Willem van der Veen, who's here as well today, who works as a spectroscopist at the NIAAA, we were able to detect ketones quite easily in the anterior cingulate cortex of the human brain in these patients. And those were primarily acetone and acetoacetate. It's more difficult at first to quantify beta-hydroxybutyrate. And the main clinical finding of this study was that those individuals who received a ketogenic diet had lower benzodiazepine intake prescribed by a psychiatrist who did not know whether people were on the ketogenic diet or a controlled diet, compared to the standard American or controlled diet that contains sugar. So people required fewer medications to have their withdrawal under control in the first week of alcohol detoxification. And also the number of people who was on a benzodiazepine decreased while on the ketogenic diet. It was lower in the ketogenic diet compared to the control diet. And every week we also did functional MRI scan where individuals were looking at cues of food, neutral cues, and alcohol cues while measuring their brain activation. And we found, we just published this actually together with Pete Menza. Thank you so much for working on this analysis. So we worked on a neurobiological trait signature that was just published last year by Hedy Kober, who works at Yale. And she developed a machine learning method based on a number of different data sets that developed a signature that predicted behavioral trading in drug-addicted individuals compared to controls, which included, among others, the medial prefrontal cortex and the ventral striatum. And what we found is that in panel B, that participants rated the alcohol cues as less, or that they wanted it less compared to the control diet. And also overall, this neurobiological trading signature was lower for the ketogenic diet compared to the control diet. And then we had a question, well, does this ketogenic diet, does it also affect alcohol consumption? And here we work primarily with Leandro Bendiscola at NIAAA, who worked with whisker rats and who first made sure they were trained on self-administering alcohol for 7 weeks. And then the rats were either receiving a ketogenic diet or a chow diet for 2 months. And below you can see again the proof of principle, ketone levels went up in the ketogenic diet group. And after that, the rats were back on chow for several weeks and were exposed to alcohol vaping. And what he found, and we published this data together, so the clinical and the preclinical, was that the rats with a history of a ketogenic diet, but currently on a regular or glucose-rich chow diet, that they reduced their alcohol self-administration. So we published all of this together in one paper, and this finding on a reduction in alcohol consumption with the ketogenic diet was found a year later also by a Spanish group, and this was in mice. And this was not a history of a ketogenic diet, but these mice were actually on the ketogenic diet when they were exposed to alcohol. And the reason why Leandro chose to look at the history was that he found an enormous effect of the ketogenic diet on alcohol sensitivity. So these rats were on the ketogenic diet for 6 weeks or on chow diet, and he exposed them to 14 hours of alcohol vaping. And it was incredible, the blood alcohol level difference between those rats who were on a keto diet compared to those on a control diet. So rats could not tolerate alcohol, or not very well compared to in the ketogenic diet group. And some of them I guess even passed out. So and we're working still on finding out what the mechanism is of this. But there's a collaboration with Volkow, Dr. Volkow, who's here. So they dissected the livers and looked at protein levels of ADH1 or alcohol dehydrogenase, which is primary in the metabolism of alcohol. And we found a reduction in ADH1 protein levels in the ketogenic diet group compared to the chow group. So this might be a mechanism as to how these rats on the ketogenic diet could not tolerate alcohol. So an interim summary on the ketogenic diet and alcohol. We performed the first ketogenic diet intervention study in humans, and this relatively rigid 4-to-1 diet was well tolerated. The diet reduced benzodiazepine intake and neurobiological craving signatures in human AED inpatients. And also the diet reduced alcohol consumption and increased alcohol sensitivity in alcohol-dependent rats. And Dr. Cindy Lee, my postdoc currently at Penn, she's following up on this latter finding. And we're setting up, or we're actually recruiting for it now, We will expose healthy human volunteers to a ketogenic diet, and we'll give them a low dose of alcohol, not 14 hours of vaping overnight, but we want to see more subtly if there are also a difference in blood and breath alcohol levels in humans. But there are many limitations to a ketogenic diet. It's pretty rigid and it's difficult to maintain. It's going to be very hard to stick to a ketogenic diet outpatient, as we also see from low retention rates in other patient populations. Then Dr. Veech developed the exogenous keto monoester, also known popularly as a ketogenic diet in a bottle. And he developed it with a DARPA grant to primarily help soldiers with their physical activity. And the first paper on human pharmacokinetics came out in 2012. And what a ketone ester basically does is when you drink it, within half an hour, Ph.D. or ketone levels are elevated to very similar levels as what we see after 3 weeks of these very, very rigid ketogenic diets. They go down again after 3 to 4 hours, though. So if you want to maintain higher ketone levels, you would have to drink this drink every 4 hours. And the first clinical paper that came out, also under Dr. Veech's leadership, was this paper with Dr. Mary Newport, who I think is zooming in. Hello, Dr. Newport. And this was a case study on her husband, and Dr. Veech actually treated her husband with Alzheimer's disease with this ketone ester, and the paper describes remarkable improvements of him, both in cognition as well as in mood. And this was published in 2015, before the ketone ester officially was in the market. So I think Dr. Veech got in trouble for this in NIAAA. But I think many people forgave him because of how astonishing the results were. So our ongoing human studies at Penn using these ketone supplements, we are studying currently the effects of ketone supplementation on brain energetics, so very similar study designs as what Dr. Volkow just presented. We also study the effects on alcohol pharmacokinetics in healthy volunteers to see, okay, what do ketones do to alcohol, and also to self-reported alcohol responses. And we have a study now ongoing on alcohol withdrawal. So basically the same study as we did here at NIH, but then with a ketone supplement. And the first study that we did has a very similar setup as Dr. Volkow's study, namely people both with and without alcohol use disorder came to the lab twice, once at baseline after an overnight fast, and once after taking a ketone supplement. And this is the same ketone ester as Dr. Veech developed from Delta G, which is now commercially available. And this is the one without sugar. We didn't want to add sugar to the mix, especially for PET glucose scans and brain spectroscopy scans that we performed afterwards. And what we found is actually a map that is strikingly similar to the acetate studies that Dr. Volkow showed. So if you drink a ketone ester half an hour before this 1 hour PET scan, the metabolic rate of glucose or glucose metabolism goes down by 22%. So so the brain, even though there's glucose in the system, seems to prefer ketones as an energy source. And this is consistent with this shift from glucose to ketone as a brain energy source when we drink these drinks. And also with spectroscopy, we looked at BHB levels. So BHB levels also drastically increase in the human brain, which again is consistent with this immediate shift. The second study that we did was in healthy, socially drinking human volunteers who again came into the lab twice. Once they drank a ketone supplement, This time we actually switched the delta G, or the Veech supplement, is terribly, terribly bitter. And this compound from Kinetic, it tastes a lot better. So our participants were very happy that we switched and didn't have to suffer from this bitter taste. It's similar. It also has 1,3-butanediol, and it elevates ketone levels, but it just tastes better. So we gave this half an hour before a moderate to low dose of alcohol, both to women and to men, and wanted to see what happened to the alcohol first. And what we actually saw that the blood alcohol levels and the breath alcohol levels, similarly to the ketogenic diet, so we were worried about that, would be much higher, such that our participants couldn't tolerate alcohol that much. But here we found the exact opposite, namely breath alcohol levels and blood alcohol levels were much lower when pairing the ketone supplement with alcohol. So it strongly blunted the breath and blood responses, and also self-reported ratings of liking and wanting more alcohol were reduced when pairing the alcohol with the ketone supplement. And then Leandro replicated this effect in breath. So we saw also with a much higher level of alcohol that they were exposed to. So about these rats were roughly would have had a breathalyzer of 0.07 or 0.06. So it's about double the amount of alcohol. And you see still a very blunted response in the Kinetic. And also Kinetic, it contains this non-caloric sweetener allulose. So we first thought that maybe that leads to these blunted effects, but no, allulose had no effect. And yeah, right now we're still trying to find out what it is. It looks more like absorption than metabolism, but we will investigate this further. So it looks a little bit as if Kinetic leads to drinking alcohol with a full meal, whereas the baseline water response, the rats and the humans had an empty stomach, but they did not. They actually ate. So. The third and last study, so this study came out in 2021, is the Denmark group again, and they compared the effects of a ketogenic diet and ketone monoester on alcohol withdrawal symptoms that they found a few years before. And they basically showed that a ketone supplement was efficient in reducing alcohol withdrawal in mice. So the study that we have ongoing now, and I don't have data yet to present, but it's basically the same study as we did here at the NIAAA, but much simpler, namely individuals with alcohol use disorder will come in for treatment of alcohol withdrawal management or alcohol detoxification treatment, will receive a bottle of Kinetic for breakfast, lunch, and dinner in addition to their regular diet. And we hypothesize, again, that the use of benzodiazepines and their alcohol withdrawal symptoms would be lower in the Kinetic group compared to the placebo group. So an interim summary on the ketone supplements. The supplement shifted brain energetics from glucose to ketones as an energy source. And in contrast to the ketogenic diet, the supplement decreased alcohol blood levels. And the ketone supplement decreased alcohol wanting and liking in healthy volunteers. So as a conclusion and future directions, ketones might be a therapeutic benefit to individuals with an alcohol use disorder by improving brain energetics and or other mechanisms, including maybe a delayed absorption of alcohol. We are working hard on finding the optimal dose and also to see which products are most palatable for longer-term outpatient interventions. We're currently very happy primarily with the Kinetic, which tastes a bit like a sour lemonade. And more studies on the effects of ketones and alcohol consumption and craving, and also on longer-term cognition in human AAD patients are needed. And they also studied the effects of the ketone supplement on alcohol consumption. So in IP or intraperitoneal and oral gavage, both of the Veech's ketone esters both led to reduced alcohol consumption. But then for longer term, it's really hard to make rats stick with this diet. They do not like it, and they lose a lot of weight. So we're working on how to improve that also in the rat study. So many thanks to my little lab at Penn, and of course to Nora Volkow for mentorship in the last 10 years, Leandro and Janina from the Skoda for all the rodent experiments, and BJ for all the help, BJ Consumption Dani for all the help with the alcohol challenge and the interpretation of these data. Todd King, the brewer of the ketone ester. Anytime when I have no idea about anything chemistry, I'll call Todd and he knows the answer. So it's incredible to work with you. And I also wanted to guide you, if you want, there is an amazing podcast with Dr. Veech online. If you want to listen to him for an hour and a half explaining everything you want to know about ketones, please Google, oh dear, what is his name? Dave Asprey. It's probably somebody well known that I don't know as a foreigner, and Dr. Veech, and you'll find this podcast. Thank you so much.
Amazingly well behaved. Yeah. So we can do two things, but I need some help. For questions in the room, we need someone to get the microphone. But online, are there any? First of all, there was a question that I believe was sent to Dr. Volkow ahead of time. Does anybody have that question? Todd, do you have that question?
Yes, the question that, sorry. Thank you. The question, we do have the firstborn from Dr. Veech's family and present in person, but the caboose for the family is hopefully online. George, if you're listening, George is the last of the Veech family. He is a physician in Portland, Oregon, practicing among their use disorder community, which is rather substantial. And he has asked whether, and this would, first of all, thank you both you ladies for the wonderful presentations. Thank you both very much for that. George has noted that with the bipolar disorder, ketogenic diet has seemed to prove or be of some benefit. And he is wondering if the ketogenic diet may be of benefit in other use disorders.
And I received the email, and Corinde said, Nora, there's this question. And so I didn't, on the top of my brain, actually, I knew that there have been studies on testing the ketogenic diet for depression. And overall, the results are showing some instances benefit and in others not. But this is not surprising per se, because one of the things that we're recognizing is that depression is a very heterogeneous condition. So understanding what are the characteristics of people that respond to a ketogenic diet, or as Corinde was showing, I think it is much more translatable, this ester could be very important because it could offer an alternative treatment. For substance use disorders, there are studies that have evaluated, and again, I Googled it, I didn't know it on the top of my brain. I knew it was not very salient because we don't have currently any grantee that's studying the ketogenic diet for other substance use disorders. But there was a couple of studies on acute administration that showed that the effects of, and I think it may have been for cocaine, cocaine were reduced during the ketogenic diet, or I think it was ketogenic diet that were given to the rodents or an ester. And that also the magnitude of the changes in dopamine produced by the drug were attenuated. But with respect to the substance use disorder, what really provided the rationale for moving it forward, and now we know that a lot from genetic studies, alcohol has a very important metabolic component, and so, and not surprising, genes that are engaged in metabolism, energetics, actually are engaged in the vulnerability for alcohol use disorder. That is not the case for cocaine or heroin. So, but that doesn't necessarily mean that it may not be beneficial. And Corinde and I were discussing it because I was asking her, well, you know, one of the hypotheses that researchers are proposing is that to increase GABA. And certainly in the brain, that could explain why it is effective for the treatment of epilepsy. But Corinde was also noting, I mean, no, not in all cases it goes. So we really do not know, and like everything else in science, sometimes you just need to try and see if there is a signal or not.
That's great.
Yeah, I think especially for mood disorders, there are not many studies that have been done that look at the ketogenic diet and the placebo diet that were blinded. So many studies suffer from placebo effect and using very small sample sizes. So it's currently still pretty unknown. There's a physician, Chris Palmer, at Harvard. He just wrote a popular book on brain energetics and other chapters on how a ketogenic diet and other metabolic interventions can help neurological conditions and psychiatric conditions. But you know, there's definitely evidence that that a ketogenic diet or ketones can help neurological and psychiatric disorders.
Okay, we have questions on the, so you can just keep the mic because these questions are from the chat. I can read them in. So I think this is Choa Sung says, or Chamandi Senavirampi says, thanks for the very interesting presentation. Have you looked at plasma glucose and hemoglobin A1C levels at baseline and during treatment?
So for the ketogenic diet intervention, we actually did not look at glucose. We only looked at ketone levels, and we were actively avoiding having glucose levels also as orders because that would unblind us most likely, because the ketogenic diet does reduce glucose levels. Then for the ketone supplements, we see huge reductions in glucose levels in the plasma. So we've had actually multiple, or that are nurse practitioners and MDs who are like, why is the glucose level of 50? What is it, milligrams per deciliter? Is this person okay? So yes, their ketone levels are incredibly high. And this was with the kinetic product, but also with the ketone ester. So glucose levels do immediately reduce in humans, not so much in rats. And so we don't really understand the difference between humans and rats of why glucose levels go down in one species and not the other. It may be somehow that ketones also have an insulin spike that simply reduce these glucose levels.
And hemoglobin, anything on that?
No, we've never.
And I wanted to ask a question to Corinde and to George, if I may.
You may.
It's actually, if you are food deprived and your ketones go up, do we know anything about how that affects that sensitivity to alcohol? So you're food deprived.
Yeah, if you're food deprived, your blood levels go up, so you're more sensitive.
You're more sensitive to alcohol?
Yeah. And you absorb more too, right?
Yeah.
So those curves that we see with ketone supplement, it almost looks as if ketone supplement leads to like a full meal.
And what I was also saying, you have been food deprived for 3 or 4 days, and you keep alcohol. What is the response to alcohol? That was my question.
I think the only studies that have been done are in fasting states, but I would imagine that was only an overnight fast and not 3 to 4 days. I'm not sure who can do that, Nora. But
an overnight, probably sufficient.
Much higher blood and breath alcohol levels.
So Bill Curtis, who is a longtime friend and collaborator, was on. Good to see you, Bill. He said, given that 1,3-butanediol has the same metabolism as ethanol, making use of alcohol dehydrogenase to make the R1,3-butanediol, what do you think R1,3-butanediol does to the levels of alcohol dehydrogenase? And in the rat, has anybody, well, you showed some ADH data, right?
Yeah, so in the ketogenic diet data in rats, we showed that the ADH1 protein levels go down, and that can explain the higher blood alcohol levels. Namely, the rats were not able to metabolize alcohol as efficiently as the rat on a ketogenic diet than on a controlled diet. But then if we give kinetic, which has 1,3-butanediol in it, we initially did the experiment because we are afraid to, if we give this as an outpatient treatment for multiple days or multiple weeks in people with alcohol use disorder, that they would overdose without even knowing. That we drink their 6-pack and suddenly it was a 24-pack, without them knowing. So we wanted to understand healthy volunteers with a low dose of alcohol, what does it do? So 1,3-butanediol did not have the same effect as a ketogenic diet. We expected that it would be competing with ADH1 on the breakdown of alcohol, but then you would expect, again, higher blood levels of alcohol. What we saw with the 1,3-butanediol or the ketone ester, substantially lower blood alcohol levels.
Mm-hmm. And there was a similar question from Dr. Newport, who also joined us. And she said, similar to Bill's question, what was the effect of our 1,3-butanediol alone on brain energy metabolism? Has this been studied? She asked because some companies are selling it apart from the ketone monoester.
No, so the only study we've done was on a ketone ester on brain glucose and brain ketone metabolism. So we have not studied, the ketone ester has both 1,3-butanediol and then BHB itself in it, but 1,3-butanediol alone is on the market by Asian men. We have not studied that. We have several studies that we still love to do. For example, is there also a competition if you give alcohol and the ketone supplements, there's competition in brain, the extra or stronger reduction in those metabolism. We haven't. I don't think other groups have done data.
I don't, but there is something that also from the data Corinde that she has found that's also very interesting, where she can actually see the very dramatic reduction in alcohol, the one that you were saying, maybe disabsorption, because that has the potential to actually be a very valuable tool when people overdose on alcohol, and they actually, you are trying to protect them from further absorption in order they don't go to respiratory depression. So her findings also have potential implications for the translation.
Yeah, that's great. So we had a couple of congratulations from Polly Joseph. That was amazing. Congrats, Nora and Corinde. This was an excellent way to inaugurate the lecture. Lucy Revo said, very interesting information, especially on the subject of Alzheimer's. Great job. But one last question from Mary Newport also. Might ketone ester be helpful to wean people off Xanax and other benzodiazepines?
We have, I mean, no one that I know of has done that study, but what benzodiazepines do in terms of glucose metabolism, it reduces glucose metabolism, but it is directly related to the decreases in energetics. So if you are looking at blood flow and metabolism, both of those parameters go down. So it has in that respect, it has very different effects from alcohol. They share that alcohol, of course, enhances GABAergic signaling, but not at the energetic level. So I do not know that it would be, but it is, like I say, sometimes these are empirical questions and someone should write it.
George, you have a question?
I have a question. So, you know, I'm listening to this and then the question about bipolar. Is there any condition that replicates what you both have shown, which is a shift from glucose metabolism to acetate metabolism in the living condition? Does chronic stress do that by any chance, where you're activating glucocorticoids? I mean, is there any other condition besides alcohol use disorder that drives this shift in metabolism? Because I find it interesting that it's such a powerful and replicable effect with alcohol. Is there anything else that drives that?
Well, it would be very interesting. I mean, and again, it comes back to the question about why are we seeing those dramatic differences? Let's forget about the heavy drinkers, on occasional drinkers. What is driving those differences? And this may pertain to your question. For example, one of the things is, what is the role of the microbiome in producing some of these alternative energetics? So could it affect it in such a way that in some individuals, the absorption of these ketones is higher just normally than someone else, and therefore they have a predominance. But it is amazing how under-investigated the whole process of energetics is overall. And I suspect that now that there's so many stories on the microbiome, this will come to emerge, including what is the role of the changes in the microbiome with alcohol or chronic consumption on some of these processes. I don't know if you have that.
Yeah, no, maybe one condition to add here, I only touched on it very briefly in Veech's case study on Alzheimer's disease. But of course, Alzheimer's disease is characterized by reduced glucose utilization, which is very similar to alcohol use disorder. But I do not think that it's not characterized by an alternative energy source. So there have been many studies done now, primarily by the group of Stephen Kinane in Canada on Keto-Therapeutics in Alzheimer's disease. And it's shown that ketogenic diet and also ketone supplements, longer term, can actually improve cognition and symptomatology as well as brain energetics in Alzheimer's. So there's a net increase in brain energetics with ketones going up, glucose going down a little bit.
But I also wonder, actually, as Corinde was speaking, my brain was running in another direction. What about diabetes? I mean, I think it's actually you cannot utilize glucose, so the brain needs to adapt. Yeah, that's actually, or I mean, when you are in insulin resistance, there is such a dramatic insulin resistance, not in type 1 where you don't have insulin resistance. Absolutely, where you will have a reduction in glucose. I suspect that esters and are keeping it. It will be interesting, and actually it will be intriguing to see, to do a PubMed and see if that's not already research, because that would be, I suspect that what would be happening.
Ben, yeah. And Paul, and yeah, that's time for a few more.
First of all, I want to thank you so much for very stimulating talks. I learned a lot. You know, I have a question when the people in myself drinking alcohol is flushing. So when I drink an alcohol, I'm typically also flushing too. So, you know, ethanol is high, acetate is high, but acetate is very low. So then when I drink the alcohol, you know, my glucose metabolism is suppressed. I don't have acetate. That causes my, you know, sleepy and alcohol. Have you, for your image test, have include anyone have flushing?
No, yeah, but
There might be
excluded. No, it was exclusionary criteria.
Yeah.
Same for us, sorry.
It's voluntary.
But it would always be unethical to give these high doses to you because the acetaldehyde would be the more toxic component. The heterozygote.
Yeah. I would say that that's something to model maybe with, you know, the kind of studies that Tommy's doing with VJ and you. You know, could we look at mice first with ALDH too? Yeah, yeah.
So it was a very nice talk, and I was particularly intrigued that ketogenic diet is actually decreasing the alcohol levels. But related to this, I have some questions. So, for example, how much this effect depends on insulin? Because whenever you drink in the pancreas, you have an insulin surge. Then you will have hypoglycemia. It will be also reflected in the brain. And then the second, after this, you probably should have like immediately like sympathogenic activation, cortisol, everything else. And for the brain, I think the most damaging is the fluctuations of the like the glucose level, metabolic level. So I predict that probably a few hours, if you would measure, your metabolism could flip to the opposite. In diabetes, also the most damaging in the cardiovascular system, the fluctuation of the blood glucose levels, not the high level by itself. I think it could be interesting also to maybe further check if you have like, because if I understood correctly, this like around between 1 hour after the top of the dose, like half an hour, 1 hour after. So it would happen like a few hours later, and you have the sympathogenic response, the cortisol response, which would all affect the metabolism. We have like very like dramatic switches in the metabolism that could be very damaging, actually, to brain or cardiovascular.
You know, one of the things that, I mean, we always had to measure the levels of glucose in plasma when we were doing the FCT because it has to be in a certain range, but we measured them before the radiotracer or close to the radiotracer administration. We didn't follow it through. And all of our studies were done approximately 20 to 30 minutes after the last consumption of alcohol. And I did wonder, and I asked myself that question, and people on an acute alcohol, how long does it take for the brain to go back to normal? This is a very important question. And no one that I have read of has done that. What we did was take people that were alcoholics and then monitor them to see how long it takes their brain to recover. And brain glucose metabolism recovers basically by 2 weeks. So by 2 weeks, you have levels that are similar to that of control. But what is the regular pattern in a normal person? And can you accelerate that pattern of recovery? And that has not been done. If I hadn't taken this job at NIH, I would have done.
We can do it. But Nora, let's actually ask also another question, because we initially wanted to give the ketones to the PET-FDG scan and maybe then give an isocaloric glucose dose and then do the PET-FDG scan. But what would lead to that? I still find it very conflicting, as if you elevate glucose levels, that does not necessarily lead to elevated levels of glucose metabolism, correct?
But it's part of the problem also of the model for the registration that we do. So you're not following, you cannot really use them.
So yeah, Paul was saying, for those of you who may or may not have heard, that measuring insulin levels will be really crucial for this, because it's affected secondarily by a lot of this.
I know it's a very good suggestion, and I actually, if you can send us any references, I would really appreciate it. Because it hasn't been in my radar.
Yeah, I only know one reference where of Brianna Stubbs at Oxford, who works with Kieran Clark, who co-invented the ketone ester with Dr. Veech, and they basically measured almost everything they could imagine after giving the ketone ester to humans, and they see this small insulin spike, but it's so small that it cannot really explain the decreases in glucose or plasma glucose.
Yeah, well, I will have to talk about this, I think, offline. By the way, Dr. Clark was there. Okay, we'll take one more question from BJ, and then we got to wrap it up. And while he's doing that, I'll say that
Thank you so
there's
much.
a lot of food back there, so people should take stuff with them when they leave. And yeah, oh yeah, yeah. There's no ketones.
Yeah, thank you so much for the wonderful talk. I really appreciate it. But you, Dr. Volkow, mentioned about the gut microbiota. But I was wondering whether you have seen any papers that the ketone body actually causes improving the gut dysbiota to treat like Parkinson, Alzheimer, or epilepsy, because of those neurological disorders are related to the gut dysbiosis. So I was wondering. And another question or comment maybe, have you looked at the alcohol metabolism through the gastrointestinal bacteria or something that's causing the change in the blood alcohol concentration?
Researchers are looking at it, and I think it's an extremely interesting area. And what makes me think, though, is we've come to recognize how crucial the neuroinflammatory processes are in alcoholism, and that alcohol is a pro-inflammatory drug. And the extent to which you actually, that some of it may be driven by the disruption of epithelial cells in the gastrointestinal tract that allow for toxins to go into the circulation that then generates inflammatory process. That, for example, could be potentially, again, purely speculative, alleviated by a diet. And I would be surprised if there's not already papers out there in the literature, because it's such an important consideration and it's potentially very relevant, but I don't have them. It's not something that I look at it. But I don't know. Have you? Sure.
No, but we're funding Craig McLean's Alcohol Center on microbiome. So I'm hopeful that we'll be getting some data, like you said, Nora.
Yeah, no, and this is, again, a new world that's coming to recognize how crucial inflammation is and how important the microbes are. So, an aspect that we haven't paid attention to. So for the ketogenic diet, absolutely. And then the question is going to be, Corinde, will these esters have the same effects on the microbiome or equivalent? And I think it's worthwhile.
I think that the only study that really jumps into my brain at this point was there was a study rat model of epilepsy, and they gave humans a ketogenic diet and translated the microbiome into the rats, and the rats were seizure-free just based on the microbiome. So that is a causal link that the microbiome alone may be sufficient for brain effects.
Well, that's great. Well, thank you, everybody. It was an amazing discussion. This was really great. So we'll be figuring out next year's speakers sometime this summer or so. So if you have ideas for candidates, please send them along to me. Thanks. That was great.
Thank you.
You know, the other thing I was thinking, though, is the ketone.
Thank you.
The ketone supplementation could be dangerous. So there's this whole idea that I had that the CIA was direction, right? Not the idea that something will stop you from living they would drink it before they drank, right?